My name is Rain Ellison. Today is April 25th, 2024. The movement of macromolecules within the cell. Cells contain many macromolecules which function systematically with one another in order to sustain the cell and ultimately the organism's life. The endomembrane system refers to the collection of membrane-bound organelles who work together to modify, package, and transport lipids and proteins. This system is comprised of 7 organelles: the nuclear envelope, endoplasmic reticulum, Golgi apparatus, lysosomes, various vesicles and vacuoles, and the plasma membrane. We will first look at protein synthesis. Starting at the nucleus, home to all genetic material of the cell, DNA is transcribed into RNA, a single-stranded macromolecule that functions in protein synthesis, gene regulation, and as the genome of some viruses. The RNA, once created, enters the ribosome. The ribosome of the cell is, in short, a protein-making factory. When the single-stranded mRNA, messenger RNA, comes into contact with the ribosome, it binds to the ribosome through a tRNA, transfer RNA, allowing the mRNA and the ribosome to work together to synthesize a protein. Proteins can function in a number of different ways, including transport, defense, signaling, receptor, contractile, structural, storage, and as enzymes. Proteins secreted by the ribosomes are then attached to the rough endoplasmic reticulum, which the ribosomes themselves are attached to. The proteins are folded into their three-dimensional shape within the rough ER, thus informing the function of the macromolecule. Following the folding within the rough ER, the proteins are then packaged into transport vesicles. Such transport vesicles then enter the Golgi apparatus, where they are sorted and prepared for transport to their respective areas inside and out of the cell. Transport vesicles have a small membrane-bound sacs in eukaryotic cells' cytoplasm, and their function is to eventually fuse with another organelle or a plasma membrane and release its contents. For example, should the transport vesicle hold a membrane protein, such as a transport protein, the vesicle would travel from the rough ER to the plasma membrane, fuse with the plasma membrane, and release the transport protein, which would then embed itself into the plasma membrane. The transport protein's role in the membrane is to allow specific larger polar ions and molecules to enter and exit the cell. Another type of protein that could be synthesized is a secretory protein. Such proteins are synthesized in a similar way as the membrane transport protein, but are then packaged into secretory vesicles rather than transport vesicles and secreted through transport proteins to be used outside of the cell in other areas of the tissue. Collagen, for example, is the most abundant secretory glycoprotein, especially in the extracellular matrix, making up 40% of the human body and serving as the matrix of macromolecules for bones. In addition to proteins, lipids are another crucial component of the endomembrane system. A lipid consists of mainly carbon and hydrogen molecules and can function in a number of different ways. In this video, we will look specifically at the phospholipid, which, following synthesis, will become part of the phospholipid bilayer of the cell membrane. The smooth endoplasmic reticulum is responsible for the production of most lipids within a cell. Following synthesis, the phospholipid is packaged in the Golgi apparatus and prepared for transport within the cell. Phospholipids are eventually bound for the membrane. Hydrophobic phosphate heads and hydrophilic fatty acid tails arrange themselves in a double layer of phospholipids such that a semi-permeable membrane forms. Lipids, like proteins, have many other forms and functions, including as steroids, glycerol, and fatty acids. Each aspect of the endomembrane system contributes to its eventual form and, subsequently, the functions of the macromolecules. These are my references for today's presentation. Thank you.